Right, we're continuing on talking about these pesky viruses, right? So with viruses and the infections that they cause, we can talk about what we call persistent infections. So persistent viruses tend to remain in the host for long periods of time, and this can be from many weeks to even the lifetime of the individual. And that would be considered to be a persistent infection, one that kind of keeps on giving, I guess we should say. Um, Now, with these persistent infections, we can describe them as either being chronic or latent. So, chronic infections are characterized by a continuous release of virions over time. It's a slow progression of a disease. So as it says, the slow, constant replication of the virus that is going on. Now, in some cases, there could be kind of a period of a quiet infection for months or even years that then can precede a much more active viral replication that happens. So in the quiet period, though, the host cells produce a small number of virions. So they're still replicating, they're still there, but the disease may not be as evident. But the idea is that they can still pass the virus on to others. And that's what you need to be mindful of. So HIV is a great example of a chronic infection. So HIV, which impacts the immune system. So this virus acts by killing T cells, and it follows this very slow progressing chronic model. So those that get infected, usually they start out in this kind of quiet stage, and they may be said that they're HIV positive because they can detect the virus, but they don't have AIDS yet. It hasn't progressed into a disease. So without treatment, though, usually the quiet stage ends and the virus counts increases over time. Now, some of these viruses that cause these persistent infections can integrate their genome into the host cell, and HIV is one of them because it's a retrovirus, right? So it goes from messenger RNA to DNA, and then that DNA becomes inserted into the host cell's genome. So this is what we call a provirus. So retrovirus are notorious for forming a provirus in infected cells. Now, some viruses, for instance, the virus that causes hepatitis B and C so hepatitis B and C do not form proviruses. So just to, kind of make sure that, that chronic infections don't always form proviruses. So some do, some don't. Latent infections. So latent refers to long periods also, but this is long periods where the virus is dormant. So it's inactive. It's not replicating. It's not detectable, but it can reactivate later. All right, so that's a latent versus chronic. You have the slow replication that's kind of ongoing in the background. Latent is it's not replicating, but it's still there. Right, it's still hanging out with you. So latent infections usually are kind of distinguished because they might have what we call flare-ups. So these would be intermittent periods where the virus is no longer inactive. So during the flare-up, it can shed new viruses, and the infected person might have symptoms during that time, and they're able to pass on the virus to others during those flare-ups. And after the flare-up, the virus kind of retreats back into being inactive again. So a lot of times flare-ups can be triggered by stress. It can be that they're sick, so they might have a fever. It could be hormonal changes. A lot of different things could potentially trigger a flare-up. So members of the herpes viridae family are notorious for causing latent infections by going dormant in certain host cells. So these viruses do not integrate themselves into the host genome, so they don't make proviruses. Instead, they exist outside of the genome. So the herpes virus usually lies dormant in the nerve cells of the host, and then reemerge to generate symptoms. So with herpes, it could be cold sores that pop up. There's also the varicella zoster viruses that cause chickenpox, and that persists in the host nerve cells as well, and then they reemerge later on in life to cause shingles. So if you think about it, kind of being inactive and then active for a short period of time and then going inactive makes it very hard for your immune system to kind of eliminate the virus. But once again, the virus can still survive and then produce lots of new viruses, and then it can be transmitted to a new host after that. Okay. So viruses that cause persistent infections are often associated with cancer. Those that can cause cancer are called oncogenic viruses. So onco referring to kind of cancer and genic for generating. So kind of cancer generating viruses. They include a number of DNA viruses as well as some RNA viruses. Some integrate into the host genome, like proviruses, while others kind of remain outside. So oncogenic viruses are thought to cause between about 10 to 15% of all cancers. And what they do is these viruses kind of stimulate uncontrolled cell growth in their host. So they stimulate the host cells to keep on dividing. Or they change the message in the host cells so that it's not as responsive to signals that call for the death of the cell, because a lot of times cells will divide a certain number of times and then die. So it may not respond to those signals, and it may keep on living and keep on dividing instead. Either way, what happens is the host cells keep on dividing. You've got more and more cells, and that's what forms tumors. So it's the virus that is affecting the signaling within the host cell and causing it to produce a lot more cells. Now, infection with an oncovirus certainly increases your risk of having cancer, but it doesn't guarantee that cancer will develop. So just FYI. One of the most well-known oncogenic viruses is the human papillomavirus, or HPV. There are over probably about 200 different types of HPV, but the vast majority cause kind of benign warts and things like that, but there's about a dozen of HPV viruses that are linked to cancer. So there's HPV 16 and HPV 18 that are responsible for the majority of cervical cancers. And we also have herpes viruses, which can be also be oncogenic. They're responsible for cervical and prostate cancers, nasopharyngeal cancers, and a few other things. Kaposi sarcomas, they are also involved with. Now, let's talk about bacteriophages. So we said viruses are capable of infecting all different types of cells, and bacteria are one of them. So bacteria are also capable of being infected by a virus. So if you think about that, we kind of hate it when the virus infects us, but think about a virus infecting a bacteria, right? We think of a virus making us ill when we get it, and will a virus make a bacteria ill when it gets it? So kind of interesting to think about, right? So these viruses specifically are called bacteriophages, so it means bacteria eating, right? So it's capable of destroying the bacteria. So in a day, or I should say in an age, where we have problems with antibiotic resistance, where we have bacteria that have become resistant to the antibiotics that we're using now, we need to look towards the future and potentially other ways to treat bacterial infections. And one way to do this, and this has kind of been around for a while, this idea, is to use bacteriophages. So these phages target bacteria. So they are very specific in the bacteria that they target. These viruses will not infect our cells because they only target bacterial cells. So if we were to use a bacteriophage as a so-called antibiotic, it would not harm our cells at all. It would only target the specific bacteria that has kind of evolved alongside of. So potentially we could use these phages as a new form of antibiotics. And the great thing about it is they evolve with the bacteria. So if the bacteria evolves resistance to the bacteriophage, usually the phage is also going to evolve a way to respond back and get back into that bacteria. Some of these bacteriophages can also produce enzymes that help to break down biofilms that can cause other types of problems. So this can be potentially good, right? Using bacteriophages to combat bacteria. All right, so I also want to talk about how these bacteriophages replicate. So we have what's called the lytic cycle. So lytic replication pathway is very similar to the animal infection pathway. So we have kind of the breakdown of those, of like the attachment phase or the absorption. So the virus and the bacteria come together just through random contact, and the bacterial tail fibers help the virus to adhere to the specific proteins on the bacterial cell wall surface. And the specificity of these interactions means that certain bacteriophages only infect certain bacteria. And that's what we have going on here. The little bacteriophage as making its attachment, right, onto our E. coli cell. That circle there is the circular DNA of the bacterial cell. So we have adsorption. Then we have penetration. So like a hypodermic needle, the bacteriophage injects the genetic material into the cell, through the cell wall, through this plasma membrane and inside. And then the empty capsid kind of remains outside of the cell. So you see the little string of DNA going into our E. coli there. Then we have synthesis. So once a viral genome is inside, the bacteriophage uses the host cell's proteins and other enzymes to kind of read the viral genes. And among the early viral proteins that get made are usually DNA aces, or enzymes that break up the host cell's DNA. So it's a little bit devious. It kind of gets rid of the host DNA. So it says like, I'm in control. Right? Then we have assembly. So once all the parts of the bacteriophage are replicated, they package up the viral genome into the capsid, and the remaining phage parts are assembled. So we have assembly coming together of all the parts, and then we have a little bit of a maturation step. Everything's kind of, once again, finalizing things, and then we have the release. So the bacteriophages encode an enzyme called lysozymes, which break down the host cell walls and cause the bacterial cell to lyse. And all these new bacteriophages are released, and they can go on to infect other cells. So that's the lytic cycle.